Hello and welcome. This is chapter 3 of our Anatomy and Physiology Cells, the living units. What are cells? Cells are basic unit of all organisms. The primary functions of a cell is cell metabolism and energy use, synthesis of molecules, communication, reproduction, and inheritance. So this is the general model of the human cell. The cell has three major parts. First, we have the cell membrane, also called the plasma membrane. We also have the cytoplasm and the nucleus. So this is the general model of our cells. Each part is labeled, and we will be talking about each part of them as the lecture goes along. So first, we will discuss cell membrane and plasma membrane. So what is it? It is the outermost component of a cell. It encloses the cytoplasm and forms the boundary between material inside the cell and outside the cell. It acts as the skin of the cell. The primary function is it encloses the cell. It supports the cell contents by giving the cell rigidity, and also acts as a selective barrier that determines what goes in and out of the cell. It also plays a role in the communication between cells. So the cell membrane is highlighted in green. It encapsulates or surrounds all the parts inside the cell. The structure of the cell membrane, the most famous one is the Fluid-mosaic model, in which it is made out of phospholipids that form a double layer of molecules. Phospholipids contain two regions: polar, phosphate-containing ends, which is the hydrophilic part, and nonpolar fatty acid ends, which is the hydrophobic part. The double layer of phospholipids form a lipid barrier between inside and outside the cell. So the fluid-mosaic model of the cell membrane, the membrane is composed of a bilayer of phospholipids and cholesterol with proteins floating in the membrane. The nonpolar hydrophobic region of each phospholipid is directed toward the center of the membrane, and the polar hydrophilic region is directed toward the fluid environment, either inside or outside the cell. Okay, so the cell membrane looks like this. We have a thick lipid layer, or our hydrophobic layer, and we have a thin bilayer of phosphate, which is hydrophilic. It likes water. So the cell membrane actually can accept both polar and non-polar substances. So this is a subunit of the phospholipid. So it has both a polar head and a non-polar tail. And this is another diagram of the cell membrane. As you can see, it is not only consisting of a phospholipid bilayer, it also contains glycolipids, some different glycoproteins, other types of proteins. So this serves different functions depending on the type of molecule. So the cytoplasm is the jelly-like fluid that fills the cell, the material between the cell membrane and the nucleus, and it is the living material that surrounds the nucleus and contain many types of organelles. The nucleus contains the cell's genetic material. So you can think of the cell membrane as the skin. You can think of the cytoplasm analogous to blood in humans, and you can think of the nucleus as the brain of the body, since it contains the genetic material. So these are electron images of our cytoplasm, highlighted in blue. So this is the cytoplasm, highlighted in blue. The nucleus is the center of the cell. You can also find mitochondria, peroxisomes, and vesicles. Next, we have our organelles, which are specialized structure or membrane-bound components within the cell that has specific functions. So this table has the summary of the organelles, where they are found, and together with their functions. Next, you have transport or movement through the cell membrane. So we were going to discuss how things move through the cell membrane. Since cells are very specific on things they can accept or excrete in the cell. So the cell membrane selectively determines what can pass in and out of the cell. Enzymes, glycogen, and potassium are found in higher concentrations inside the cell. And sodium, calcium, and chloride are found in higher concentrations outside the cell. Please take an important look at these parts. This will be asked. The movement may be passive or active. So first, let's discuss the passive membrane transport. It does not require the cell to expend energy in the form of ATP. That is why it is called passive. It requires no metabolic energy from the cell but solely relies on physical properties of the substance themselves. We have different types of passive membrane transport. We have diffusion, osmosis, and facilitated transport, or facilitated diffusion. Next, we have active membrane transport. It does require the cell to expend energy, usually in the form of ATP. So it has different types: active transport, secondary active transport, endocytosis, and exocytosis. So this is the summary of the different parts of the different types and characteristics of movements across the cell membrane. Okay, so to discuss diffusion and osmosis, let us rediscuss what is a solution. So a solution is made out of solute and solvent. So a solution is a homogeneous mixture composed of two or more substances. A solute is a substance dissolved in another substance, which is the chemical dissolved in a solution. And the solvent is the substance that dissolves a solute. So this is it. Diffusion can be defined as the net movement of particles from an area of high concentration to an area of low concentration. Diffusion is movement of particles, particularly this is the movement of solute, from high to low. It can be viewed as the tendency for solutes, such as ions or molecules, to move from one area of higher concentration to an area of lower concentration than solute in solution. So tendency of smoke or perfume to distribute throughout the room, dye or salt throughout a beaker of water. So in any case that you experience putting in food color in water, it does not stay in one place. What happens is the concentration of the food color in water tends to equally distribute throughout the water in the container. And that is diffusion. So diffusion works on a concentration gradient. So the gradual difference in concentration of dissolved substance in solution between a region of high density and one of lower density. Okay. Next, we have osmosis. So osmosis is diffusion of water, a solvent, across a semi-permeable membrane, such as the cell membrane, from a region of higher water concentration to a region of lower water concentration. It relies on osmotic pressure, which is the force required to prevent the movement of water across a selectively permeable membrane. The measure of the tendency of water to move by osmosis across a selectively permeable membrane. Okay? So the greater concentration of a solution, the greater its osmotic pressure, and the greater the tendency for water to move into the solution. Okay? So what is the concentration of the solution? Most of the time, we will use the concentration of sodium chloride. Okay? % sodium chloride. Okay? So where there is a higher concentration, and we have lower concentration of our sodium chloride. Please take note that it's not actually just sodium chloride, but in physical chemistry, when discussing tonicity, we like to convert every substance into an equivalent amount of sodium chloride. Okay, water, we are talking about water, and there is a semi-permeable membrane. Water will go from a region of lower concentration to a region of higher concentration. Okay, so if we had a laboratory experiment and we use blood has certain amounts of sodium chloride. If we just put water, plain distilled water, water has no sodium chloride, it will enter the red blood cell and cause it to explode. Not explode, but to swell. Okay, so we have different types of osmotic solutions. First, we have our hypotonic solution. It is the lower concentration of solutes and higher concentration of water than the cytoplasm of the cell. Comparatively, a hypotonic solution lacks the concentration of the cytoplasm. The solution has less tone or osmotic pressure than the cell, and water tends to move into the cell, causing swelling or rupturing after a while. Next, we have isotonic solutions. Isotonic solutions have a similar concentration to the cytoplasm. Nothing happens in this solution, and the cell neither shrinks nor swells. And we have our hypertonic solution. This is a solution in which the concentration is higher than what is inside the cell. Since we are talking about osmosis, since the solvent outside the cell has higher concentration, the water inside the cell will move out of the cell, causing the cells to shrink. Okay, so this is a summary of what happens to different solutions when you put blood in them. Okay, so as a medical professional, what you need to know is we should always use isotonic solutions when infusing into humans. Next, we have our active transport mechanisms. Another passive transport mechanism, which is our carrier-mediated transport mechanism. So this relies on the movement of transport of large water-soluble molecules or electrically charged ions across a membrane by means of a carrier module. That is why it is called a carrier-mediated transport. It exhibits specificity, meaning one carrier can only carry one type of molecule. It is specific to one type of molecule. These are transported by carriers. We have different kinds of carrier-mediated transport. A carrier-mediated transport process that moves substances across the cell membrane from an area of higher concentration to an area of lower concentration of a substance does not require ATP. And this is called your facilitated diffusion. Okay? Take for example that facilitated diffusion of glucose. Since glucose is found higher outside the cell, it will be transported inside the cell, which has a lower amount of glucose. Next, we have active transport, a carrier-mediated process that moves substances across the cell membrane from regions of lower concentration to those of higher concentration against a concentration gradient. So this is high to low, low to high. And this requires ATP. Okay, take for example, this is the sodium-potassium pump. Sodium is pumped inside the cell, while potassium is pumped outside the cell. Okay, since sodium is less is so this transport requires ATP. Next, we have secondary active transport. This involves the diffusion of transported substance such as sodium down to its concentration gradient that provides the energy to transport a second substance such as glucose across the cell membrane. It is called co-transport, in which case both substances move in the same direction. In counter-transport, both substances move in opposite directions. Okay, take for example, we have the sodium-potassium pump. So this is what confused me earlier. So sodium goes out while potassium goes in. This is the opposite, or our counter-transport. While sodium and glucose going into the cell is co-transport, meaning they both get inside the cell. Next, we have our endocytosis. Endocytosis is the movement of materials into the cell by a formation of a vesicle. So a vesicle you can think of is a pouch. The process of cellular ingestion by which the plasma membrane folds inward to form a vesicle containing the material to be taken into the cell. We have different types. First, we have our phagocytosis, which is cell eating, in which solid particles are ingested. And we also have our pinocytosis, which is cell drinking, the intake of liquid particles. Okay, so this is how it happens. We have receptor-mediated endocytosis, which is the movement and transport of a substance into the cell after binding with the receptor molecule. So the molecules bind to a receptor and it slowly forms a circular shape. As it fills the shape with molecules, it will slowly close and it will separate from the cell membrane, and this is called your vesicle. Next, we have our exocytosis. It is the opposite of endocytosis, in which it is going outside the cell. It is movement out of the cells by vesicles. It allows the release of materials from the cells and secretion of materials from cells by vesicle formation. So a good example is secretion of digestive enzymes by the pancreas, secretion of mucus by the salivary glands. So this is the opposite. A vesicle forms inside the cell membrane. It slowly reaches the cell membrane and it opens up in the cell membrane, releasing what is contained in the vesicle.